In 2015, David joined Protein Data Bank in Europe, and at the European Bioinformatics Institute. And then in 2018, he took a role of outreach and training lead of PDBe. And over to you, David.
Great, thank you very much, Maria, for the introduction. And thank you for the speakers for inviting me to talk. So yes, I'm from the Protein Data Bank in Europe, and I'm outreach and training lead. But I'm also part of the curation team at PDBe. So within our team, we work on curating data that's been deposited to the PDB and to the EMDB as well. So to begin with, I'm going to talk about a bit about recent developments in deposition, and then move on to talking about some of the impact of that deposition data and the additional impact that it can bring. So firstly, a bit of background on data deposition to the PDB, and I guess to start with what the PDB is. So the Worldwide Protein Data Bank is a consortium of various different sites across the world. So there's the RCSB PDB in the US and PDBj in Japan, and ourselves at PDBe are based just outside Cambridge in the UK. And along with EMDB and BMRB, we form the Worldwide Protein Data Bank, and we all work together on the deposition of data to those core data archives. So the map here shows the geographic distribution of how these entries are deposited. As you see, everything from Europe and Africa will come to PDBe and be processed by our team. But we do all these things in collaboration with with one piece of software that we all share, and that all goes into one single PDB archive at the end. So the deposition we use is called OneDep. So this is a single shared deposition system across the wwPDB. So the benefit of having one shared system that we all work to develop and improve means that we can standardize the data in the PDB, so we can ensure we're getting consistent data coming in, and that we're not missing information across too many different entries. So as you can see, there's various processes that go on in the curation process of PDB data, which is what our team are involved in doing. So we take information about the small molecules, ensure that we give unique IDs to ligands. We process sequences and map the protein sequences to UniProt. And there's also standardized validation processes. So I won't go into too much detail about the process of deposition here, but we do have a useful guide if you are interested in smoothening the process of deposition to the PDB or EMDB with these 5 steps to deposition. So if you want to find out more, you can visit wwPDB.org and view more information about the steps that are required for a deposition. I want to talk a bit more about what are some of the new things that we've integrated recently into OneDep to try and help benefit both depositors and the community. So one of these new things is an assembly review page within the deposition interface. So previously, after curating an entry, we'd send the entry back and give a description of the curated assembly, but now we've integrated Molstar as a 3D viewer into the deposition interface so that depositors now have a visual representation of what that curated assembly is. And this makes it much easier for depositors to then review the assembly, confirm this, or communicate with us if there are any problems with that assembly. And the benefit of this is this should hopefully improve the curation of assemblies in the archive. And we use that assembly information for so much more when we put this out on the public websites. And so having accurate assembly data is really vital for that process. One of the other improvements we've had last year was to support multi-datablock structure factor files. Now this was in collaboration with the wwPDB MMSIF working group, and we've extended the MMSIF dictionary for structure factor files to create a richer description of that experimental data. And it also benefits because we can bring in more quality metrics as well alongside the reflection data. So a lot of this work's been done in collaboration with global phasing and the MOSIF working group, as I've mentioned. So it does enable archiving of results of STAR and ISO. And you can also directly deposit structure factor files to OneDep from Autoproc, Buster, and Gemi that has all this information, and it should ensure that all this information goes straight through to the PDB archive without causing too many problems in terms of back and forth between our curation team and depositors. And it was also mentioned, I think earlier in this study weekend, that it will soon then be implemented in CCP4. Because these extensions are standardized across the dictionary, these can then be implemented by any refinement softwares. So some of the other updates at wwPDB include planning for extended identifiers for both PDB and ligand IDs. So we are starting to run out of codes now. Back in 1971, when the archive first started over 50 years ago, I don't think we thought 4-character codes would be running out anytime soon, but we're getting there now. And so we're working on, we have a 8-character new code that's prefixed by PDB that will replace this in time. We're still issuing 4-character PDB IDs. However, in the archive files themselves, we're also adding these extended codes. And so hopefully this is something that the community can begin to implement ahead of time, so that when that time does come, it's not so much of a shock. One of the other changes we've made is to make PI data public for new depositions once they're released. So this includes the name of the PI, their email address, and an ORCID ID. And the reason for this is that, particularly with the COVID-19 pandemic, we were seeing a real increase in the speed of release of structures before there were necessarily publications available where the author data was available. So because of this, we've added this change so that for release structures where they're currently unpublished, there is a medium for people to get in contact, the community to discuss structures with the depositors, and hopefully to improve these structures and generally improve the archive as a whole. So I'm going to talk briefly about some of the new methods for generating models for molecular replacement, but I'm going to do it briefly because this is something which I think we've been over quite a lot in the last couple of days, and some of this Gerard has very recently gone over. So it seems a silly thing to say there's not enough structures in the PDB, but although there's 55,000 distinct proteins around and about in the PDB archive, if we compare this to UniProt, this is really quite a small proportion of kind of the known protein structures. So this is where these structure predictions are becoming really helpful because of their increase in accuracy in trying to provide alternative structures to be used for things like molecular replacement, but also other research. And just a note, of course, being from the wwPDB, of how important the PDB archive has been for creating training sets for this data, and I think how important it will continue to be. And as Harold mentioned, we will just continue to improve and increase the number of structures in the PDB thanks to these improvements. So as Harold mentioned, our team at PDBe have been heavily involved in the development of AlphaFold DB, the collaboration between EMBL-EBI and DeepMind. And the key goal for 2022 is aiming to have all these UniRef 90 proteins in the database. So this is a reference archive, and the idea being that once we have all these in, there will actually be a structure prediction for everything within 90% similarity of a known protein UniProt, which obviously for something in the realms of molecular replacement is fantastic. We're also, as the PDBe team, working on improvements to the search features and other features on the website as well throughout the year. So to keep an eye out for changes on that as well. Again, we've been working very hard on 3D Beacons network as well. Again, Harold mentioned this, so I won't spend too long on this. Just to point out that this is very much an open collaboration, that we want to bring as many different resources that provide protein structures, whether these be experimentally determined or predicted, and bring them under this 3D Beacons hub so that people can easily access them. So there's various ones that Harold mentioned. I saw Robbie put in Slack that AlphaFill is also going to have a 3D Beacon as well. Also Model Portal is due to join as well. So we're hoping that this continues to expand, and this provides a place for easy access of protein structure data from a whole host of resources. So we're encouraging all these increased depositions. We've got all this new data coming from predictions. What can we do with it? Well, we've been working on our Protein Data Bank in Europe knowledge base resource. And one of the key things with this is we're trying to answer scientific questions with structural data. Now, one of the main things from this perspective is that more than 90% of PDB data users aren't structural biologists. This is something that Harold mentioned with the invent of AlphaFold as well. We're going to have just a larger number of non-structural biologists who want to access this data. So these are some of the questions they might want to ask: how the various structures of proteins differ, which small molecules interact with those proteins, and also shedding light on the effects of variants on structure as well. So we're trying to enrich that data through PDBe and try and put the structural data in a biological context. So by bringing together all these different resources under one umbrella, and this is very much a community-driven resource, we should hopefully provide more context to relate to this data, but also provide kind of a standard access to all this information and make it easier for people to find it, particularly for non-structural biologists who might not be aware of all these different tools and resources to support their work. Now, the first iteration of pages from PDBe are our aggregated views of proteins. So obviously, traditionally, we view PDB entries as individual entries, single structures. The idea of these pages is we change that focus from being a single structure to being a specific protein. In this case, focusing on a uniprot accession. And then we can provide all the related structural data for that given protein. Initially, we've done this through PDB structures and all the data we have within the archive. We've also added functional and biophysical annotations as well from PDBe partner resources so that can be integrated alongside structured data. We're also integrating through 3D Beacons as well, access to structure predictions as well, so that comes under that as well. So this data is presented in these interactive visualizations that we've had to develop to try and display this data in a way that's easy for people to digest, particularly in 2D sequence views, so that for biologists coming to look at this type of data, they can easily see large amounts of information summarized in a concise way. But we have expanded to more 3D visualization tools in these aggregated views, particularly with the superposition process, where we have proteins superposed against all of the PDB structures in the archive for that protein. And that allows us to identify conformational variability and cluster ligand binding sites as well and see this visually. So I'm just going to go through a couple of examples of where I think these pages really help to tell the story of the science behind it as well. So for example, if one of the questions is related to ligand binding and how this might affect protein conformation. So this example is adenylate kinase from E. coli, which as a kinase catalyzes transfer of phosphate, in this case from ATP to AMP to create 2 molecules of ADP. And so initially from the PDBe pages, we know there are 17 different structures in the PDB with 9 unique ligands bound. And I pointed out 3 of these particular ligands. So there's AMP, of course. ANP in this case is an analog of ATP. And then we have AP5, which is kind of a conjoined ligand of 2 adenosyl with the phosphates in between. And if we look at the sequence view to the bottom here, let me just get my laser pointer to highlight it, we kind of end up with two clear binding sites. We have for the AMP here, these patterns, and for a NP, we have an alternative binding site. And as would be expected for this kind of paired ligand AP5, we have both binding sites occupied. But we can also view the superposition of these, and it shows that there are two distinct clusters that are formed for this particular protein. And one of those clusters in green is an open conformation, which is the unbound conformation, while orange is the bound and closed conformation, where ligands are bound, and that, you know, inherently creates a closed conformation of the protein. We can also see from superposition and looking at the ligands that are bound that the open conformation is also found when we only have an ATP analog without AMP bound. So it shows that both ligands are needed to form this conformational change. And just the final example of something that we can't really do with classic PDB entries is show things like fragment screening, because there's so many PDB entries involved in the fragment screening experiment, and these are split across hundreds of pages. So the example here of the main protease from SARS-CoV-2, by viewing this in the aggregated view, you can actually more easily overview this data to see all the interacting residues from the 2D viewer here and see where those interactions occur, but also from the superposition, and you can see where some of these fragments are clustering in the binding site, and use this as a stepping stone, like all these pages, as a stepping stone for the next stage of the research. So just one final thing is to just discuss the future plans for PDBe-KB. So we want to try and change the way that people are looking at this data. So this initially has begun with us looking at these aggregated views of proteins. What we want to try and do next is move to aggregated views of ligands, where people who are interested in a small molecule ligand can come and see where all the similar ligands, what protein targets do these bind to, and are there any common interactions found between those? And these pages we're actively working on at the moment. And also the aggregated views of complexes. And we've been working with our colleagues at Complex Portal to try and identify complexes, so that rather than having to come in from a protein perspective, people can ask a question about a complex and say, yeah, what are the various conformations of the complex? Are there clear stoichiometries for how it's formed? And how maybe certain ligands binding affect it. So I'm going to leave that there and just acknowledge obviously all our wwPDB partners and everyone we collaborate with, and of course our funding bodies. And thank you for listening. And if you do have any questions, please ask them through Slack and I'll get to those.